Okay, we can probably get started. Let me introduce you. Hello, everyone. This is Jiming San. I'm the host for AI for Health webinar, and this is our number 27. And so today, I'm thrilled to welcome Professor Connor Coley, Associate Professor at MIT in Chemical Engineering and also Electrical Engineering and Computer Science. Connor's research sits at the intersection of AI and chemistry, where his group developed models to understand and design molecules with a focus for a therapeutic discovery. And his work has earned numerous honors from NSF Career Award and Forbes 30 Under 30 Award to MIT Teaching Award and recognition, and as a Schmidt AI 2050 Fellow and Samsung AI Researcher of the Year. So beyond MIT, he also advised several AI-driven discovery companies and served editorial boards of leading journals. And on a personal note, I actually first heard Connor's talk right before COVID, if I remember, at MIT. There was this, I think it's a machine learning drug discovery summit, and I think it's in 2019, and his talk was wonderfully clear, both in the concept of machine learning and also the chemistry side. So I remember how much I enjoyed it. So I'm sure today our audience and students will find the same inspiration in his presentation. So without further ado, let's please join me welcome Professor Connor Coley.
Excellent. Well, thank you, Jimang, for the very kind introduction and for the chance to share some of our work with all of you today. And thanks everyone for listening. So what I'm going to do today is talk about a couple of the recent projects we've been working on over the past year, sometimes extending a little bit further back, applying generative techniques to small molecule design. And of course, this is in the context of small molecule drug discovery, sort of in support, of course, of human health. Now, briefly, just as a research group, I wanted to say that we work on a range of topics at this intersection of AI and chemistry. Much of our work has to do with AI for synthetic organic chemistry. We'd like to think about models to understand chemical reactions and predict the products of reactions, design synthetic pathways, and so on. Part of our work also deals with analytical chemistry and problems like computational metabolomics. So thinking about ways to take sort of messy spectral data and infer what kinds of molecular structures are present in complex mixtures. We sort of do this by making contributions both to sort of machine learning level, sort of thinking about new architectures, new tailoring techniques for adapting architectures from off-the-shelf models. But we also like to keep a sort of eye on the application side. And so we're really sort of application motivated, I would say, in much of what we do. And so one of the sort of main third categories that we work in, which will be the focus of this time together, is thinking about the use of AI for medicinal chemistry and designing molecular structures as drug candidates in sort of early stages of either hit to lead optimization or other sort of early stage optimizations. And so we do this mostly for small organic molecules. There's a range of different modalities, of course, that are used as therapeutics. We like to still work with small molecules where you benefit from thinking about them in terms of their atomistic sort of constituents. So we don't think so much about the design of proteins. There's plenty of other wonderful work in the field for that. We like to think about small molecules and maybe going up to macrocycles, which you can often view as just large small molecules. So these are the kinds of structures that we think about, and we think about the computational strategies that can facilitate their design and discovery. And so at a high conceptual level, right, the way that we arrive at these kinds of solutions as approved medicines, or even in use in agriculture and material science and other applications, the way that we arrive at these is really as some iterative optimization problem. And so we like to frame molecular discovery as a search or an optimization. Right? It's a multi-objective optimization. You might think about Pareto optimization strategies. But nevertheless, regardless of how exactly you define your objective, you can think about this goal-directed search through chemical space to try to find molecules meeting your property needs. And in the simplest description, we're going to be iterating through these design cycles of proposing candidate structures, making them, testing them experimentally, and then using sort of any experimental data that we get to revise some models, which will hopefully inform then the next round of design. So there's ways of using sort of AI and ML techniques piecemeal within this sort of framework of an iterative discovery cycle. And there's ways of using AI to also unify across multiple stages. And we'll see a little bit of that in the discussion today. Now, the design part is what we're going to focus on. This is one of the most important questions in a molecular discovery campaign. And one way to think of it is basically just what do we do next, right? Given the information that we have at hand, given the information that's accessible to us, what kinds of molecular structures are going to be most worth making and testing experimentally? There's a couple of main paradigms for doing this. People have been trying to understand the relationship between molecular structure and their properties for a long time. This is sort of a cornerstone of computer-aided drug design, building quantitative structure-function relationships that we might draw as these cartoon surfaces where the molecules that exist at these peaks have better properties and the ones in the troughs have worse properties. And so the optimization task might be to find the molecules that live at these peaks, right, at these local maxima, or hopefully the global maximum. Now, given that we can build these kinds of models to anticipate properties with some fidelity, we can use virtual screening approaches to sample chemical space, propose hundreds, thousands, millions, billions, or trillions of chemical structures, sort of sampling across what we believe to be sort of valid small molecule structures, sort of arrangements of atoms and molecular graphs. And we can simply try to predict or anticipate their properties and maybe use these property prediction models or simulations to tell us which ones are better and which ones are worse. But the promise or the allure of generative models is that perhaps we can navigate this landscape more efficiently, and we can use different generative techniques to learn to propose the structures that live at these maxima. And again, in the most idealized view of the world, do some sort of hill climbing to find some molecular structures with the best properties without the sort of uniform sampling of virtual screening. And so I'll talk just really, really briefly about some of the work that we've done in virtual screening, because this does still serve a major role in early stage drug discovery, but we're going to then move on pretty quickly to generative modeling. And so for virtual screening, what I'll focus on is a very simple optimization strategy that we can apply to virtual libraries. And what are virtual libraries in the first place? Well, these are collections of compounds that chemical vendors often are willing to list and then are willing to sell us. They're proprietary databases that we also use as virtual libraries. Most pharmaceutical companies will have internal databases of chemical structures that they think they can make. And that description is actually really important. So these libraries are most effective when they are defined in terms of what chemicals do we believe to be synthetically accessible. And so when you go to a company that sells you these molecules like enamine or wuxi, they will try to list out all of the different product molecules that they think they could access through a series of nice, well-defined, robust synthetic steps. And so just by enumerating what products do you think you can make given the reactants you have in your inventory, you can easily reach into the billions and trillions and more of hypothetical chemical structures. So using commercial building blocks and reaction rules, we can start to enumerate these virtual chemical spaces. However, these spaces start to get very large very quickly, right? It's this combinatorial explosion of different options. And so no matter what you're using as your property predictor, especially if it's some sort of simulation technique or a crude simulation technique like docking, you can't really afford to explicitly evaluate trillions of structures. And so a very sort of simple strategy that you can do is take a subset of your space rather than the full exhaustive library and evaluate those, use the results to train a surrogate model and iterate. So this is relying on the fact that it's pretty easy and cheap to train a surrogate model and to use that surrogate model for inference on a very large chemical space, at least compared to docking in this situation. So rather than evaluate 100 million candidates in a brute force manner, we can instead evaluate a few 100,000, train a model, and that model will tell us where in chemical space to look and where in chemical space not to look. And so this works fairly well. The relationship between structure and docking score is somewhat smooth because docking scores are very, very crude approximations of binding affinities. They correlate, but not terribly well. And so what we can do is we can save lots of computational time, often orders of magnitude, using these surrogate models to help us navigate these larger libraries. So from an ML perspective, this is just a discrete optimization using a variety of different surrogate models. They all work just to different extents. So what else can you do with this sort of basic framework? Well, you can do this optimization for a single docking score, of course. You can also do Pareto optimization to understand the trade-offs. And to the extent that you can anticipate off-target interactions through docking, which again is a little bit questionable, we can explore the trade-offs between multiple targets and find the molecules that live on Pareto fronts. We've also added in some additional sort of bells and whistles to try to speed up this process by sort of fathoming entire regions of chemical space. So in a similar way to how, branch and bounds techniques will prune entire portions of a search tree of your sort of feasible sets, we can use uncertainty quantification to tell us which parts of chemical space are unlikely to ever be viable, and can we just sort of discard them entirely and not revisit them in future iterations. And we've also been working on different techniques to try to improve the diversity of sampling. So coming up with different strategies of defining diverse batches of molecular structures where that diversity is not based on just sort of chemical structure, but it's really based on what a model believes and looking at the covariance between predictions in a model's posterior. So this has been sort of interesting work trying to build on this way of accelerating virtual screening in this context of virtual library navigation. But as I already mentioned, the focus today, I want to be on the generative side of things, right? The hope and the promise is that we don't need these large libraries of millions, billions, and trillions of structures. We can simply use generative techniques to propose the candidate structures that will have the properties we want. That's the dream. The basic idea for most generative techniques, not all, is that you have some database of chemical structures. These might be just sort of compounds that have been made before, compounds that have biological activity. And you use these to teach a generative model what normal molecules look like. What's the distribution of sort of unconditional generation of molecular structures? Then you can start proposing new structures. And if you have an evaluator providing feedback, you can start steering generation towards molecules that have particular properties. And so you still do need this evaluator in many cases. But once you have it, you can steer direction in a bunch of different ways. This is just an animation showing the results targeting a predicted degradation behavior for PROTACs. And so the surrogate model predicting this behavior is, of course, highly imperfect, but it steered our generative technique, in this case, graph invents, towards a structure that resembles very much a PROTAC. Now, there's some problems with generative models, right? There's a reason why they haven't, solved everything in early stage drug discovery. And one of the reasons is that their creativity in sort of proposing new structures that free us from predefined chemical libraries can also serve as a detriment. And so sometimes we see that the kinds of structures these models propose are a little bit too inventive, right? They come up with motifs that don't make a lot of chemical sense. These are structures that pass the sort of basic cheminformatics validity rules that our tools impose, but they are not reasonable structures at all. And different models sort of exhibit this failure mode to different extents. But this has really been one of the reasons why I think generative modeling has not fully lived up to its expectations that were set out now almost a decade ago. So again, going back 8 or 9 years, the promise was that we could harness things like the language of chemistry expressed through smile strings, and then use the power of text generation models to propose these new structures that would help us find more potent drugs faster. And what we've encountered, and what's been quantified through numerous benchmarks and anecdotal studies, is that most of the structures that are proposed by these techniques tends to not be synthesizable. Many of the algorithms used to optimize for different properties are not sample efficient. They might require hundreds of thousands of attempts, which is not sort of matching what's feasible to explore experimentally. And just in a practical sense, a lot of these models aren't that good at meeting the property criteria that we're establishing. So they're not great even at optimizing docking scores. And a lot of the molecules that are proposed by structure-based models, which we'll talk about soon, don't actually achieve as many of the favorable protein interactions that we see in good, strong selective binders. And so two of these limitations, right, synthesizability and not achieving good interactions, will motivate the rest of this talk. And we're going to talk about two threads of work that really try to tackle these different failure modes of generative models. We're going to start with the latter, thinking about how to condition molecular generation on particular targeted interactions. Now, we started this work thinking initially about shape. Shape is a very powerful tool for virtual screening, right? In a lot of ligand-based applications to do ultra-large sort of library searches, people will use shape as a measure of similarity to find what ligands might be similar to a reference ligand or a reference hit molecule that's appeared successful in a phenotypic screen. So they're very popular techniques, very fast techniques like ROX and ROX color that are used to do these quick similarity searches over large chemical libraries. And so our work in this space started with the question of how can we recapitulate this, or can we recapitulate this kind of behavior with a generative technique so we can condition generation on a target 3D shape rather than doing this sort of exhaustive querying of a reference database of structures. And so the goal here was to devise a technique where we could specify, here's the shape that I want for a small molecule structure, and we could then generate in 3 dimensions the molecular structure that will approximately occupy that shape. So the first model that we sort of built along these lines is called SQUID. So this was sort of using this nice sort of equivariant encoding of the molecular shape. We were then also embedding the molecular graph. We were using this sort of variational way of training the model so that we could then decode diverse molecular structures that shared shapes. And the decoder for this model, which we started, circa 2022, a little bit before, was this autoregressive sort of fragment generation approach. And so we would grow molecular structures fragment by fragment, sort of selecting attachment points and dihedrals to attach as we would gradually fill in this target volume that we've specified in this conditional model. And so we were able to train a model that could then generate chemical structures that matched target shapes. We could also tune the degree of chemical similarity. So we could decide whether we wanted to sort of sacrifice shape similarity to have more chemical diversity. Right? From a drug discovery perspective, you might want to hop to diverse scaffolds to maybe free yourself of other liabilities of the molecule, because of course there are plenty of non-shape reasons why these molecules might look better or worse. And so given this sort of target reference molecule, you can generate a conformer, express it in this blue mesh, encode that, and then decode different chemical structures that fill this volume approximately equally well, that do express very different functional groups and sort of different motifs there. And so SQUID, again, was the first step to thinking about using shape and 3-dimensional considerations as a primary sort of conditioning during molecular generation. And this proof of concept did show that we could use SQUID to generalize across many different types of conformation. So if one reference structure was very flexible, we could encode many different conformations of that reference and decode many different analogs that could recapitulate those shapes. And if certain shapes were more or less important for that molecule's performance or efficacy, we could sort of find the structures that were most responsible there and pursue those analogs preferentially. And so I keep describing this as sort of a proof of concept or a first model, and that's because it worked well at accomplishing what it was designed to do, but there were a couple of really key limitations. So the first key limitation of this model was that generating molecules fragment by fragments worked, but it was very clunky and sort of very difficult to train, just sort of empirically. And I think, based on where generative techniques are now, right, the sort of autoregressive fragment growing sort of dates this model for folks in the audience who are familiar with this general field of work. The second limitation is that shape is, of course, not really the goal, right? Shape is used as a proxy often for the kinds of interactions ligands can express. That's why rocks work so well. But really what we're after is thinking about the interactions that a ligand can achieve with a protein of interest, right? In sort of a classic single target way of thinking about drug discovery. So binding between a small molecule and a protein is a function of many, many things. You need some basic level of shape complementarity. You need to have some sort of electrostatic complementarity, but you also quite often rely on these selective pharmacophoric interactions, things like hydrogen bonding, different sort of like pi-pi stacking. There's a wide range of different ways that medicinal chemists sort of express and describe the favorability of different binding modes through these non-covalent interactions. On top of this, there's of course this whole other layer of flexibility, but that's not something that we're going to touch on in this talk. So there's this rich vocabulary of interactions, and for folks who are interested and don't come at this from the domain side, I'd encourage you to check out this really nice journal article in JMedChem about sort of the whole suite of molecular interactions that can and do influence binding. It's a very comprehensive discussion of these. So these kinds of interactions are known to be important. They're discussed by medicinal chemists. They drive binding. They influence selectivity. And if you look at the sort of set of 3D protein structure conditions generative models that exist, or at least that existed a year or two ago, many of them could generate molecular structures that achieved some degree of shape complementarity. And if you were to sort of redock the compounds using docking tools, the scores were often quite favorable. But there's this nice article, nice benchmark called PostCheck that just highlighted that despite passing sort of benchmarks on a number of metrics, if one looked at the number of hydrogen bonding interactions and other non-covalent interactions between these generated ligands in a protein, typically the generated structures displayed far fewer than the ones that were either in known binders with co-crystals, or in particular in this cross-docked data set, which is sort of designed to have these kinds of interactions. And so we wanted to sort of take these interactions and take the knowledge that these interactions are desirable and bring them back into generative modeling itself. And so we can really just extend or think about extending that conditional generative framework from just shape to now being shape plus the full set of interactions that a ligand might express. And so at a basic level, we can include electrostatics, and we can include those pharmacophores that I mentioned, like hydrogen bonds, donors, and acceptors. And so our idea is to learn the joint distribution of molecular structure and these interaction profiles, we call them, so that we can do this conditional sampling, given the interaction profile we want, generate diverse 3D molecules, new structures, new scaffolds. That was sort of the goal with this shepherd model. Now, to learn this sort of complex joint distribution, we turned to diffusion modeling. So again, I think many folks in this audience won't need a primer, but of course, diffusion modeling really sort of completely took over image and video and many other generative modalities. The notion that you can learn very complex distributions by training a model to iteratively denoise a sample, of course, later largely supplanted by flow matching and other techniques. But this idea of learning complex structures was readily extended to molecules by folks other than us in the field, right? Demonstrating that just like you can remove noise from RGB pixel values, you can denoise 3D coordinates and atom types of atoms, then assembling full molecular structures. And so you can teach models to generate new three-dimensional structures that obey valence rules. They mostly obey sort of have decent strain energy. And so this notion of using diffusion modeling for 3D molecular design has been well established by this point. Also, conditional sampling using sort of diffusion modeling has been well established, in painting being one of the main techniques for doing so. There's many different ways of using guidance during sampling, but in painting is arguably one of the simpler ways of doing conditional generation. So again, in pixel space, in painting is masking out certain regions and then trying to generate a sample that's consistent with the pixels that we are preserving in the query, sort of filling in these diverse ways of imputing or sort of extrapolating from what we're conditioning on. And so we're going to use that for molecular design by learning, again, to in-paint sort of molecular structures conditioned on this interaction profile, because we've learned this sort of full joint distribution over those sort of 4 different modalities or representations. And so that's what Shepard does. So Shepard is a model that learns that joint distribution over 3D molecular structures, the solvent accessible shapes they exhibit, the electrostatic surface of points on that surface, as well as the discrete set of pharmacophores. The nice thing about learning this joint representation over these sort of 4 different channels, if you will, is that we can train Shepard purely on ligands. We're going to apply it to drug design, but we are not going to rely on the existence of ligand protein poses, co-crystals, or any experimental data like that. We're simply going to generate as much training data as we want from ligands in isolation, because we can easily derive from them what their electrostatic surface would be, what kinds of pharmacophores they can express. That's sort of using traditional cheminformatics approaches. So we represent 3D structures in sort of the normal way that you would in this field. We have atom types, we have formal charges, we have positions. We also sort of track covalent bonds. We represent point clouds for the solvent accessible shape, just as a point cloud. We have sort of relatively high density of point sample to have some shape fidelity for the structure. Electrostatic surfaces just need to assign a scalar charge, effective charge, to each of those points. And then pharmacophores, we're going to represent those as these typed vectors. So pharmacophores have discrete types, right? It's very different if you're a hydrogen bond donor versus acceptor. So we need to encode the type, we need to encode the position. But things like pi-pi stacking relies on the directionality of how those aromatic rings interact, cation-pi interactions. There's a lot of directionality that becomes relevant. And so we also need to have that notion of an orientation. And so pharmacophores are represented as these vector clouds, essentially. That's how we encode these interactions. I already mentioned that the way we do conditional sampling is through in-painting. So we train this model to learn that joint distribution. We just build it on top of Equiformer v2. And then what we can do is we can sort of denoise all the channels together, replacing the interaction profiles of the shape, electrostatics, and pharmacophores with the ground truth we're trying to condition on to get that molecular structure. So with this model, we can do some really nice ligand-only things. So we can take ligands from the PDB, we can extract out the structure, we can re-optimize its geometry so we don't know the real pose, we just know the structure of that molecule. And we can use Shepard to generate analogs of that molecule that are predicted to have similar interactions, again, in terms of the shape, electrostatics, and pharmacophores. We can take the generated structures, redock them, and see that they do have similar interactions, at least to the extent that docking can perceive them. So even though we've had sort of no knowledge of the protein pocket, no knowledge of the binding pose in this model, we can simply do this hit expansion of these reference structures to find new molecules that recapitulates key aspects of their interactions with the binding pocket. Beyond the sort of hit expansion, we can also do interesting things like fragment merging. So in fragment-based drug design, one can take protein crystals, soak it with a lot of very small structures. You end up doing sort of that crystallography to figure out, okay, where might the footholds be for binding of ligands. You typically look at these sort of messy superimposed structures where all the fragments that docked and sort of found their way into any of these different sort of grooves are superimposed. And you can see, okay, here's the general area where we might be able to get a good ligand. So from the fragment screen, which here is the superposition of 13 fragments, we can extract out what are those interactions in terms of what pharmacophores do those fragments exhibit. We can then use that as the basis of sampling from Shepard, right? Use that as the conditional profile. And given that conditional profile, again, we go through this sort of denoising process using in-painting for conditional sampling. And then we end up with sort of one of our recommended ligands that we can then take and again, redock sort of resuperimpose onto those fragments to get a sense of how it might bind. So we've done a lot of computational work with Shepherd, validating its use cases. We have some ongoing experimental work trying to validate it prospectively. That's, of course, where the real proof is going to be. But we've seen sort of all signs pointing to the fact that Shepherd can do this really nice recapitulation of interactions while achieving diverse scaffolds and letting us sort of navigate to different chemical series. And one of the sort of areas where Shepherd doesn't do quite as well as we would like it to is in trying to analog natural products. So this is an area where you might think that, oh, I have a very complex natural product. Maybe this has some good, interesting biological activity, but this is not a very developable compounds, right? It's difficult to make analogs. It's really difficult to work with. It's expensive. It's tedious to synthesize. And so you might imagine a model like Shepherd being used to generate analogs that can recapitulate some of the key interactions that these natural products express in smaller, more compact structures that hopefully might preserve their biological activity. And so we did see to some extent Shepherd doing that. We got okay sort of similarities in recovering the kinds of interactions we might want to, but you can see there's sort of still a lot of complexity in a full, larger natural product that's hard to recapitulate with a smaller structure. So this is something that we're still sort of thinking about. You can condition on part of these structures instead, and maybe that's going to be another way of looking at analoging. For this final demonstration for Shepherd on trying to take natural products and convert them into functionally similar, simpler analogs that we can then develop more easily is a good segue into the sort of second part of this talk, sort of other threads talking about synthesis-constrained molecular generation. Because one of the main reasons why you wouldn't want to directly work with this natural product is because it's very costly to synthesize, derivatize, and so on. So how might we operate in a synthetically tractable chemical space instead of that very complex space? That's the sort of motivating question for the sort of next thread of work, which I'll go through now in less time than the first. We already saw this slide that showed a bunch of unrealistic structures that can come out of generative techniques when you don't really think about synthesis or stability at all. There's many reasons why some of these molecules are unrealistic. Synthesizability is just one of those reasons. And so we wanted to ask, now dating back many years ago, this is 2019, 2020, how can we basically mitigate this failure mode and not have generative techniques give us these pretty wacky structures that we could never take into the laboratory and test? We've worked on tools for organic chemistry, as I mentioned at the very beginning. This has led to a variety of sort of open source software for synthesis planning. And so we can just take the designs that come out of generative techniques and subject them to retrosynthetic analysis. We can very rapidly perform retrosynthesis to understand, is there a simple synthetic pathway to reach these structures, or might they be more synthetically difficult than we want to deal with? And so we can use tools like retrosynthesis tools that are now out there in open source to help answer that question. What we found is that sometimes this filters out everything. So it could be the case that none of the molecules that are generated make it through this screen. Now we can also introduce softer penalties during molecular design. So there are a bunch of heuristics for synthetic complexity that can be incorporated. And so you might imagine that instead of optimizing just for something like a docking score, we bias that by some synthetic accessibility heuristic, again, of which there are many. Or for different sort of generative techniques, there might be ways of ensuring that the biased generation doesn't deviate too far from that initial chemical space of what reasonable molecules look like. And so this is what reInvent, a very popular tool from AstraZeneca, does. You can control the extent to which the generative structures will deviate from the prior, which has been trained on sort of fairly reasonable looking structures, I think in PubChem in their case. But instead of these sort of soft constraints, which encourage but don't enforce synthesizability, we've been trying to advocate for using rigid constraints. So instead of having generative models propose molecular structures through string generation or graph generation or 3D point cloud generation or any sort of structure-focused description, we can instead ask them to generate new structures through a reaction-focused description. So we can force our generative techniques to basically live in synthesis space, to propose molecular structures through the proposal of a synthetic pathway. In practical terms, this looks like constraining the generative technique to use predefined building blocks and predefined reaction templates, or otherwise sort of adhering to what we understand to be the rules of chemistry. So one of our earlier approaches to this was effectively a tree generation task. So in a bottom-up manner, we can train a policy to select commercially available building blocks, choose graph or tree editing actions like adding on to this growing tree, merging two different trees together to come up with a full synthetic pathway that encodes a product molecule. So this is essentially just some hierarchical policy network that we can condition on a target molecular embedding. We can train this on known molecules with known synthetic roots, and then we can apply it to optimization just by wrapping this with a simple genetic algorithm. That works fine. So we can change that conditional code. That's going to change the decisions made by the policy, giving us a new synthetic route, a new molecule, new properties. Again, that's very conducive to any sort of model-free optimization technique. And the punchline of sort of moving to this synthetically constrained way of doing molecular design is that for the same objective function, we can end up finding chemical structures that are far simpler than what other generative techniques would propose. So every molecule that comes out of this model is forced to be associated with a synthetic pathway, sort of stemming from commercially available building blocks that we constrain it to. And that leads to these very small, very simple recommendations compared to sort of fragment-based approaches or atom-by-atom approaches that are very, very effective optimizers, but they can optimize an objective function at the expense of chemical reasonableness. And so there's sort of strengths and weaknesses of both kinds of techniques. But if your goal is to have synthetically tractable structures and things you can immediately go into the laboratory and test, thinking in terms of chemical synthesis has these pretty strong benefits. Now, this version of the model, as I mentioned, was sort of a tree generation model. It worked well. It wasn't the most scalable, I would say, architecture in terms of pre-training on very, very large numbers of structures. Now, one of the sort of pivots came when Shitong Luo was working on sort of similar techniques for synthesis-constrained generation, in this case, proposing the use of postfix notation as the way to encode synthetic pathways. So rather than actually treat synthetic trees like trees, we can treat them like sequences, just by linearizing these sort of operators and operands that we have in the route. And of course, this is with direct analogy to postfix notation used in other sorts of, program contexts, sort of stacking up different, again, operands and operators to encode these sort of implicit computational trees or computational graphs. And so postfix notation lets us take a synthetic tree and turn it into a sequence. And once you can express things in sequence form, then of course, you can use extraordinarily scalable architectures for sequence generation. So here we can sort of just pull the transformer architecture and use it for the generation of synthetic pathways through the iterative decoding of building blocks and reaction templates. And so this model, we can use the same constraints as the first one. We can force it to use commercially available building blocks. We can force it to use these sort of expert-defined reaction templates that we can encode and curate by hand. And we can then generate, in a much more scalable way, these synthetic pathways. So this model had two versions. One was an encoder-decoder architecture. One was a decoder-only architecture. You can then apply reinforcement learning to for optimization. But in the encoder-decoder architecture, one of the really nice things that one can do is take not just reference molecules that we know to be synthetically tractable to decode their pathways, but take molecules that we don't know to be synthetically tractable, or we know not to be synthetically tractable, and then decode them. And so in this way, as Chatang describes it, right, we can project from chemical space onto this manifold of synthetically accessible structures. So this encoder decoder takes any molecular structure as input and decodes then a synthetic pathway to a synthetically accessible analog of that. And so this is in some ways a nice byproduct of the design of the model. It lets us go back to those kind of funny generative techniques that make mistakes and propose unrealistic structures, and now have ways of cleaning them up. So we can take designs that don't make a whole lot of sense, and we can come up with synthetically tractable analogs of them that are associated with a pathway. We can go into the laboratory, we can test them readily. We can take these motifs that are a little bit suspect, we can in this case expand the ring into something that's much more reasonable, again, corresponds to a known building block fragment we can then go into the laboratory and test. And there are, of course, going to be exceptions to how well we preserve properties and scores, because we are requiring the generative molecules to abide by our building blocks and our templates. And so there's some simplifications that need to be made and some sacrifices that might occur in terms of performance. But again, we guarantee that every molecule that's proposed is associated with a synthetic pathway connected to commercial starting materials. So it keeps these very, very actionable, and empirically does seem to preserve the scores fairly well. So we showed this also for binding modes, looking at synthetically tractable analogs in 3D, redocking them. We tends to preserve most aspects of the binding mode of the original reference structure. And so this line of work, right, coming up with ways of ensuring that generative techniques are operating within the space that's accessible, constraints to building blocks we can buy and reactions we know how to run, is very practical. And it also should sound very familiar, because this is exactly the same set of constraints that were in those make-on-demand libraries, right? This is sort of how companies like Enamine list out the billions of molecules that they're willing to sell you. They're doing the same kind of constrained enumeration of products. And so the key difference here is that rather than having this explicit combinatorial enumeration of products, and then ending up with this long list of products to search against and query, we're instead keeping all of that implicit chemical space inside of our generative model, right, which knows how to navigate this vast, vast space of trillions plus different structures. But when we apply it in an optimization setting, this generative model only samples the molecules that it thinks it needs to on the way to finding the best structures. And so we gain all the benefits of these large virtual libraries in terms of proposing structures that we know how to make, but we don't have to enumerate them, and we have all the convenience of a generative technique that lets us iteratively explore this candidate space. We can go a little bit deeper in terms of the number of reaction steps. We can go a little bit broader in terms of the number of building blocks. So there's a few other practical benefits as well, but if nothing else, we are sort of getting the best of both worlds, right? The best of deep learning models to do iterative optimization and the practicalities of these virtual libraries. So to sort of summarize what I've hopefully conveyed today, we think a lot about molecular design as a key part of how AI is used in early stage drug discovery. Some of this work does build very directly off of existing virtual screening workflows. Much of it is thinking more in the generative space, but even in the generative space, we're really trying to ensure that these algorithms and these methods are practical, and we're trying to mitigate some of the failure modes that we've seen other techniques suffer from in the literature. And so for Squid and Shepherd, that we're starting to think about shape condition generation and then interaction conditions generation, so that we can have ligands that achieve sort of higher rates of non-covalent interactions, hopefully being more potent and selective as a result. And then in sort of the last half of the talk, thinking mostly about how we can constrain generative techniques by synthetic tractability as a way of hopefully fully replacing these make-on-demand virtual libraries, so that we don't have to enumerate these spaces in a brute force manner, but we still preserve all of the benefits. So hopefully this has been sort of an interesting way for you to see how we think about tailoring different AI models to this domain of chemistry and ensuring that these tools are practical and usable. And with that, we'll just sort of acknowledge the group, acknowledge sort of generous funding support. And in particular, we work with a large number of pharmaceutical and chemical companies through the MLPDS consortium, who've always been a very sort of good set of partners in helping inform the direction of these tools and ensure that we are actually solving some of the problems that we're setting out to solve. And with that, thanks very much, and looking forward to any discussion.
Great. Thanks, Connor, for the wonderful talk. So for the audience, please, if you have a question, you can ask the question directly or put it in the chat. And so maybe while we're waiting, I have a question maybe to kick off, Connor. So as you know, we have been working on this clinical trial space for some times. And recently, FDA released all this complete response letters from recent trials, I think maybe within the last 10 years. And our group have been analyzing those letters, trying to understand, okay, what are the... So for maybe, I'm not sure you know those letters, but they pretty much describe what went wrong for the trial. Right? So I thought, okay, probably the major issue will be efficacy, safety, right? That's pretty much the top two things that clinical trial is trying to study.
Mm-hmm.
Apparently, the biggest categories of issues are chemistry, manufacturing, control related. I was very surprised. And then when I asked around for the folks who work in the pharma space, they said, oh, yeah, that's known to be the main issues for a lot of the trials that didn't get approved or have some issues with their manufacturing processes. So I don't know if you have some thoughts in terms of you have been using AI for helping finding those molecules. Do you think those manufacturing type of, I guess, metrics or issues can be addressed a priori? So
Yeah, no, it's a good question. So I think the short answer is because the value of working at those manufacturing challenges is so high by the time a molecule gets to that stage that there's a lot of eyes on the problem, a lot of experts working on it. And so I think likely the challenges that are faced are sort of on the more advanced side of things that might be more difficult for these models to help with at the current state. But we do think about how, right, AI can help plan synthetic routes, not just for discovery chemistry, where you're trying to make milligram quantities and you don't care about cost so much, to process chemistry, where you do need to think about, what does my impurity profile look like? What's the sort of toxicity of byproducts? What's the cost of goods manufactured? And so we do think about, and we're increasingly thinking about how do we use these techniques to help design the more robust, scalable routes to make these molecules. There are a lot of complexities in doing that, and it's not going to be something that is simply solved by the machine learning side of the equation because this is not a data-rich regime. The considerations that go into manufacturing are often very subtle. Each molecule almost feels like it's its own, special case. And so I think there's going to be sort of a combination of maybe computational chemistry approaches and some data-driven approaches that might help our ability to anticipate those challenges and help overcome them. So it is something that we're looking into. I hadn't seen that particular sort of analysis or those results that led to that particular conclusion, but certainly we think about process chemistry as an open challenge for AI systems beyond just synthesis planning here.
makes sense. So maybe a small follow-up on that. So a lot of this molecule generation property prediction tasks seems are predicting very, I guess, pretty far away from this clinical endpoint, right? Whether the drug
Yep.
works or whether they will have some biological clinical effects. Do you think that's a more of a challenge for gathering that data? That's why people are not trying to predict that in this stage, or is
Yeah,
specific things that's okay? Why we're only predicting docking score or some kind of a binding affinity, right? Not something more downstream.
I do think that a lot of it comes down to sort of access to that data and that data being seen as sort of appropriate for benchmarking. I mean, there's a lot of issues with how people benchmark property prediction models in general. There's been sort of different benchmarking efforts that have sort of come and gone over the years, and there's the therapeutic data commons. So there's the Polaris benchmark, which is gaining a lot of traction. But I think predominantly, right, the data sets that people have viewed as common molecular property benchmarking data sets have not included quite as much clinical data. And I think in part it's access to that data, but I think in part it's probably access to any relevant training data and the ability to get good predictions. So it's not something that we have personally worked on in the group. So Iambic, though, a company that I advise, has had some pretty good success using large language models, sort of ingesting vast quantities of less structured data and some structured data to predict these more clinical. But I think it's not something that's the maybe academic fields that do property prediction benchmarking have paid as close attention to.
Mhm.
And it might just be awareness and access, I guess.
Okay, makes sense. Cool. Any questions from the audience? Hello.
Can I ask a question?
Yes, Zifeng, go ahead.
Yeah. Yeah. Hey, Dr. Coley, thanks for your talk. So I think a lot of people, like including us, we are like kind of interested in building AI scientists, like driven by AI agent to perform like some tasks. Like in our example, we perform like trial design, trial optimization, and so on. I think quite a few also interesting, like making molecules using large language models and so on. So I see all of those works you have introduced. They are like kind of very highly sophisticated AI models optimized for specific like molecule specific task, like predicting property, generating molecules under some constraints. So how do you see like an large language model or an AI agent can play the role in integrating those tools together to like orchestrate them to perform a more downstream task like identifying or designing a therapeutic candidate for disease biomarker and so on. So how do you see they can perform the complementary to each other in the system?
Yeah, I'm much more a proponent, I think, of tool usage than of using like an LLM directly for something like molecular generation. I think there's a lot of proofs of concepts of a lot of things, but I do think that there's merit in using more structured tools as individual components still, based on the performance of what's currently out there and what's been demonstrated. I mean, any of these constrained models, for example, are things that could easily be integrated as sort of generative tools. You could rely on LLMs more to orchestrate the evaluation and sort of critiquing things as a panel of medicinal chemists would, looking at these on a whiteboard or on a screen. Um, so I think any of these models are sort of amenable to being ingested as and used as tools by sort of agentic workflows. I think that there's been a lot of interest, of course, in using LLMs to replace these, I'll call them more narrowly scoped tools. And again, I think there's plenty of proofs of concept that you can do that. And often the demonstration is that you don't lose performance by significant amounts, but you don't tend to gain performance. But I do think that LLMs have different priors and different biases that might be helpful. So for example, nothing that I presented involved a surrogate model for predicting tox. And maybe that's the kind of thing that just by virtue of having read the vast literature and ingested data sets of toxicity, an LLM could feasibly score just as well as a little graph neural network that you trained on a structured data set. But I think on the generation side for molecular structures, I feel like using these as tools makes more sense than generating structures directly from an LLM. But that's a little bit of a personal bias, I think.
Yeah, that makes sense. I think that's also common practice, like using AI models like as tool calling for the agents. But
Yeah.
I do recently find a challenge, like because those tools they are all optimized in separate data set or optimized for their own targets. But when you apply an AI agent working with all of these tools, the AI agent is set as a target that may not be directly related to the objective of all the individual tools optimized for. So by far, we don't see an efficient approach to optimize this agentic AI system as a like a whole system together for objective, but all those tools keep frozen or like there's not easy way to like optimize them together. So do you see any opportunities or like any possible way to make this happen, like in optimizing them together for a broader scope task with AI agent tools?
Yeah, I guess it probably depends on the tool, like whether one needs to retrain them or refocus them. I mean, the way that I see our generative techniques, at least, is that they're effectively designed for black box optimization given any oracle that takes in a structure and returns a score. This third category, the synthesis constrained ones. And so that's not the kind of thing that you would need to tailor to a particular goal or target because that oracle would serve that role for you. I mean, there are other ways. So Philip Schwaller has done a lot of work in sort of more general conditioning of generative models with some more structured or and some less structured ways of doing that conditioning and guidance. But I feel like for us, a lot of our methods are designed to be agnostic to that task. And so it would hopefully be able to be ingested and not need to be sort of retuned towards each different objective.
Yeah.
Cool. Thanks, Connor. I know we're kind of on top of the hour. I usually ask this question for some advice for our students, especially you work with very diverse set of students, right? Probably some from chemistry background, biology background, and some are coming from CS, machine learning background. And instead of tell them, okay, this is the skill you should develop, I bet you have some criteria, right, from the sample of amazing students you have developed. Like, okay, these are the type of trade that I'm looking for from my students, right? If maybe that would be a good advice for our students to think about if they want to get into the AI for drug discovery space, they need to develop that trait. Do you have some suggestion on that?
Yeah, I think for me, one of the number one things that I look for and that I admire in students and postdocs that exhibit this is the sort of motivation to identify what are the practical problems, like what are the reasons why these tools aren't being used more widely in industry, right? Why hasn't this problem been solved before? And trying to focus on addressing those issues and those failure modes and not necessarily chasing after benchmarks that are maybe well established, provide easy paths to conference papers, but getting a better number on that particular benchmark doesn't mean that a particular pharmaceutical company is going to benefit from this method. So we really like to stay grounded in our conversations and interactions with collaborators in industry and definitely members of the group who also share that interest, I think, end up with a really good perspective on problem selection as a result. So I guess that's my generic recommendation is the sort of, work on problems worth solving, not just the ones that will give you a conference paper.
Yeah, that's definitely great advice. I think especially for our computer science students, I felt because of the volume of publication in all those conferences, sometimes students feel very eager to have to publish, right? This is like it seems the shortcut, like do this benchmark and have some small incremental changes that will get me a new RIPS paper. That's the path I would take, right? But what you were saying is really think about why we're doing this, right? What's the practical impact that this can lead to that will have much longer term impact, right? Okay, that's a great advice. Thanks, Connor. I think this is amazing talk, and I think our students will enjoy and learn a lot from this. And thanks again for your time and